Hello students, in the previous video we studied about C3 pathway. Now in this video we are going to study about C4 pathway. Okay, so the C4 pathway, C4 cycle is also called as cooperative pathway. Cooperative pathway because it occurs due to the cooperation between two types of cells, that is mesophyll cells and bundle sheath cells. Cooperative pathway. And this pathway is also called as Hatch and Slack pathway because the details of this pathway is given by two scientists. They are Hatch and Slack. Hence it is also called as Hatch and Slack pathway. And it is also called as dicarboxylic acid cycle. Dicarboxylic acid cycle because the first carbon dioxide fixation product is oxaloacetic acid, which consists of two acid groups in it. Hence it is called as dicarboxylic acid cycle. So it is also called as dicarboxylic acid cycle as the first carbon dioxide fixation product, that is OAA, is with the two carboxylic groups, acid groups. Hence it is called as dicarboxylic acid cycle. So here I am making the structure of this oxaloacetic acid. Which consists of two acid groups. So this is one acid group and this is another acid group. Hence it is called as dicarboxylic acid cycle. So for the first time, a scientist named Kostak, he observed that in sugarcane plant, he observed that there is formation of oxaloacetic acid, which is the first carbon dioxide fixation product, which is C4 acid. Hence this pathway is called as C4 pathway. And the plants which exhibit this pathway are called as C4 plants. But the entire pathway, the details of entire pathway is given by these two scientists, Hatch and Slack. Hence it is called as Hatch and Slack pathway. And one more important thing is the plants which exhibit this pathway consists of special type of anatomy, leaf anatomy, that is kranz anatomy. So kranz anatomy is nothing but presence of bundle sheath cells. Kranz anatomy, that is presence of bundle sheath cells around the vascular bundles. Thick walled bundle sheath cells around the vascular bundles is called in the form of a wreath around the vascular bundles, that is called as kranz anatomy. Let's see how this kranz anatomy is exhibited by the leaves of these C4 plants. Okay, so I am making the epidermis. This is upper epidermis of the leaf, which is also called as adaxial epidermis. So this is adaxial epidermis. This is abaxial epidermis, which is lower epidermis of the leaf. Abaxial, lower epidermis of the leaf. Right. At the center, I am making a vascular bundle. Vascular bundles, where they are present? They are present in veins. So this is the vascular bundle. Vascular bundle consists of xylem and phloem. So xylem is present towards adaxial surface of the leaf, that we know. So this is xylem. I am making xylem here, and this is phloem, which is present towards abaxial surface, abaxial, lower, abaxial surface of the leaf, lower surface of the leaf. Right. Okay. Now around these vascular bundles, what are present? Special type of cells called bundle sheath cells, which are thick walled cells. They are present around the vascular bundles in the form of a wreath. Hence, this anatomy is called as kranz anatomy. So, these vascular bundles can be present in several layers, and they can extend up to adaxial epidermis and up to abaxial epidermis, that is towards the lower surface of the leaf and towards the upper surface of the leaf. And these bundle sheath cells are also associated with the mesophyll cells, which are present nearby to them. So these are the mesophyll cells which are present nearby to these bundle sheath cells. Okay, now let's study the differences between mesophyll cells and bundle sheath cells which are involved in performing this C4 pathway. So C4 pathway is cooperative pathway which occurs due to the cooperation between two types of cells, that is mesophyll cells and bundle sheath cells. These are bundle sheath cells. Now let's study the differences between mesophyll cells and bundle sheath cells. Right. Okay, now differences between mesophyll cells and bundle sheath cells. I am writing mesophyll cell as MSc and I am writing bundle sheath cell as BSc. Right. Okay, mesophyll cells consist of granal chloroplasts. Granal chloroplasts. Granal chloroplasts means chloroplasts which are with grana in them. They are called as granal chloroplasts. Whereas bundle sheath cells consist of agranal chloroplasts. Agranal chloroplasts. Okay, so granal chloroplasts now, what will be present? Granal chloroplast means thylakoid membranes are present in chloroplasts which are stacked one above the other in the form of piles of coins, forming a structure called granum. This is one granum. So here in grana, what is present? Both non-appressed regions are present. In the previous videos, I explained what is non-appressed region and appressed region. So please go through those videos to know about this appressed and non-appressed regions. Okay. So in the granum, non-appressed regions are present. This one and this one is non-appressed region and this is appressed region. So what is present at non-appressed region? PS1 is present. At appressed regions, what is present? PS2, photosystem 2 is present. So what can we say now? In case of the chloroplasts which are present in mesophyll cells are granal chloroplasts which consists of both photosystem 1 and photosystem 2 in them. As they consist of both photosystem 1 and photosystem 2 in them, both non-cyclic ETS occurs, that means non-cyclic photophosphorylation occurs, and even cyclic photophosphorylation also occurs. That is cyclic ETS also occurs. So as non-cyclic photophosphorylation occurs, what happens? This chloroplast requires external electron donor. What is that external electron donor? Water. Water which splits into 2 protons, 2 electrons and nascent oxygen. That means oxygen is released inside the mesophyll cell. Now let's study about bundle sheath cell. So in case of bundle sheath cells, the chloroplasts present are agranal chloroplasts. So now what can we say? We can say that C4 plants consists of, they exhibit dimorphism with respect to chloroplasts. That means C4 plants consists of 2 types of chloroplasts. They are granal chloroplasts and agranal chloroplasts. Granal chloroplasts are present in mesophyll cell, whereas agranal chloroplasts are present in bundle sheath cells. Right. So agranal chloroplasts, that means these thylakoid membranes are not stacking. They are not stacking one above the other. They are present singly. So here only non-appressed regions are present. There is no appressed regions. Only non-appressed regions are present. Appressed regions are absent. As there are only non-appressed regions, what is present? Only PS1, photosystem 1 is present. But photosystem 2 is absent due to the absence of appressed regions. As there is only PS1 is present, there is no occurrence of non-cyclic ETS. But there is occurrence of cyclic ETS in case of the agranal chloroplasts of bundle sheath cell. So as there is no non-cyclic ETS, there is no requirement of external electron donor. So there is no water splitting and hence there is no release of oxygen. Right? So these are the major differences between mesophyll cell and bundle sheath cell. And I want to give you some more details about this bundle sheath cell. Okay, so these bundle sheath cells are larger in size when compared with the size of mesophyll cells. Bundle sheath cells are larger in size when their size is compared with that of mesophyll cell. Okay, bundle sheath cells. These are large sized cells, when compared with that of mesophyll cell. And one more thing is they are thick walled cells without any intercellular spaces and hence intercellular spaces are absent. Intercellular spaces, I am writing as ICS, are absent. Next. And these cells, as they are thick walled cells without any intercellular spaces, they are impervious to gaseous exchange. That means they does not allow the exchange of gases, that is into the bundle sheath cell or out of the bundle sheath cell. So they are impervious to gaseous exchange. Impervious to gaseous exchange. So there is no exchange of gases. Gaseous exchange does not occur, like oxygen or carbon dioxide. They cannot enter or they cannot exit through the walls of this bundle sheet cell. Okay, and they consist of large number of chloroplasts. Number of chloroplasts present in these bundle sheet cells are more when compared with that of the number of chloroplasts present in mesophyll cell. And the size of these chloroplasts is larger when compared with the chloroplast size of mesophyll cell. So this is about some details of bundle sheet cell and mesophyll cell. Now let's start with C4 pathway in detail. So before explaining about the details of this C4 pathway, I want to give you some details of Rubisco and peptase enzyme. Two enzymes, that is Rubisco and peptase, carbon dioxide fixing enzymes. So Rubisco and peptase. Right. So this Rubisco enzyme is present in C3 plants, C4 plants, and even in CAM plants also. Whereas peptase is present in C3 plants. In C3 plants, this peptase is absent. In C4 plants and CAM plants, this peptase is present. Okay. In C3 plants, this Rubisco enzyme is present in mesophyll cell. In C4 plants, this Rubisco enzyme is present in bundle sheet cell. And whereas in CAM plants also, this Rubisco enzyme is present in mesophyll cell. Now coming to peptase. Peptase, anyway, it is absent in C3 plants. It is present in C4 plants. In C4 plants, it is present in mesophyll cell. Whereas in CAM plants also, it is present in mesophyll cell only. Okay. So now in case of C4 plants, what can we say? In bundle sheet cell, Rubisco enzyme is present, whereas peptase enzyme is absent in bundle sheet cell. In case of mesophyll cell, in C4 plants, peptase is present in mesophyll cell, but Rubisco is absent in the mesophyll cells of C4 plants. In case of CAM plants, CAM pathway occurs only in one type of cell, that is mesophyll cell, which consists of both Rubisco and peptase. So this about this CAM pathway, I'll be explaining about the next video, about this CAM pathway in the next video. So this is all about Rubisco and peptase. One more difference is this Rubisco is chloroplast based enzyme. Whereas peptase is cytoplasm based enzyme. That means it is present in the cytoplasm of mesophyll cell. Whereas this Rubisco is present in the chloroplast of mesophyll cell or bundle sheet cell. Next, coming to another difference between this Rubisco and peptase. Rubisco is involved in fixation of carbon dioxide and even oxygen, depending upon the conditions present inside the cell. What are those conditions? Those conditions are, if the concentration of carbon dioxide is more than oxygen inside the cell, that is at the site of this enzyme, what happens? It leads to carboxylation, which leads to photosynthesis, which leads to the occurrence of photosynthesis. If the concentration of oxygen is more than the concentration of carbon dioxide inside the cell, it leads to oxygenation reaction. That means it functions as oxygenase, which leads to photorespiration, the pathway called photorespiration, which is a wasteful process. Next, coming to peptase. Peptase is involved in the fixation of only carbon dioxide. It is not involved in the fixation of oxygen. That means it functions as only carboxylase but not as oxygenase. So when it functions as carbon dioxide, it leads to only what? Photosynthesis, but not in photorespiration. It is not involved in photorespiration. So these are the differences between Rubisco and peptase. Now let's start with the details of C4 pathway. Okay, let's study C4 pathway in detail. So the C4 pathway occurs in two types of cells, in cooperation between two types of cells, that is mesophyll cell and bundle sheath cell. So this is mesophyll cell, and this is bundle sheath cell. Bundle sheath cell. Right. Okay. So at first, atmospheric carbon dioxide enters into mesophyll cell through stomata, and inside the mesophyll cell, this carbon dioxide reacts with water, resulting in the formation of carbonic acid, that is HCO3 minus. Here I have written this pathway for 6 molecules of carbon dioxide. So 6 molecules of carbon dioxide enters into mesophyll cell through stomata and reacts with 6 molecules of water, resulting in the formation of 6 molecules of carbonic acid, that is HCO3 minus. Now this HCO3 minus is fixed into PEP, that is phosphoenol pyruvic acid, which is 3 carbon compound, resulting in the formation of a product called oxaloacetic acid. This oxaloacetic acid is C4 acid. Okay, as the first carbon dioxide fixation product is oxaloacetic acid, this pathway is called as C4 pathway or C4 cycle. Right. So this carboxylation, that is carbon dioxide fixation into PEP, is catalyzed by an enzyme peptidase, that is phosphoenol pyruvate kinase. So this phosphoenol pyruvate kinase is peptidase. Phosphoenol pyruvate kinase. This phosphoenol pyruvate kinase is a strong carbon dioxide fixing enzyme. Why it is strong carbon dioxide fixing enzyme? Because it can fix carbon dioxide present inside the cell, inside the plant cell, even under low concentration of the carbon dioxide. Hence it is strong carbon dioxide fixing enzyme. And one more thing is, it functions as only carboxylase. That means it leads to only photosynthesis. Whereas Rubisco functions as both carboxylase and oxygenase. But here it functions as only carboxylase. That means it is involved in the fixation of carbon dioxide into PEP. So this is a strong carbon dioxide fixing enzyme. Right. Okay. Now this oxaloacetic acid undergoes reduction into malic acid. So this step of reduction is catalyzed by an enzyme malate dehydrogenase. So here for reduction, what is required? Protons and electrons are required. These protons and electrons are donated by NADPH. So NADPH, 6 molecules of NADPH undergoes oxidation into NADP plus protons and electrons. These protons and electrons are donated to, transferred on to oxaloacetic acid, and hence it is reduced into malic acid. Right. Now this malic acid, which is formed inside the mesophyll cell, is transported into bundle sheath cell. Both of this bundle sheath cell and mesophyll cell are connected together with cytoplasmic connections, that is plasmodesmatal connections. These gaps are plasmodesmatal connections. So through this plasmodesmatal connection, malic acid is transported into bundle sheath cell. Inside the bundle sheath cell, now this malic acid undergoes oxidative decarboxylation. That means here oxidation is taking place and decarboxylation is taking place. So it undergoes oxidative decarboxylation into pyruvic acid, which is C3 acid. So when oxidation occurs, breakdown occurs, what happens? Protons and electrons are released from malic acid, which are picked up by NADP plus, and hence NADP plus is reduced into NADPH plus H plus. So that is oxidation. So here malic acid undergoes oxidative decarboxylation into pyruvic acid, whereas NADP plus is reduced back into NADPH. So there is no net utilization of NADPH. Right. So as it undergoes decarboxylation, here carbon dioxide molecule is released. So from 6 molecules of malic acid, 6 molecules of carbon dioxide is released. The 6 molecules of carbon dioxide is fixed. It enters into C3 pathway. It is fixed into RUBP, that is ribulose 1,5-bisphosphate, by the activity of Rubisco enzyme, which is RUBP carboxylase oxygenase, which leads to C3 pathway that occurs inside the bundle sheath cell. We already discussed now that Rubisco enzyme is present in bundle sheath cell, whereas peptidase is present in mesophyll cell. So here C3 cycle occurs. By the end of C3 cycle, what will be formed? One molecule of glucose, that is hexose sugar, is formed. So here there is an important point to note that whether the plants are C3 plants or C4 plants or CAM plants, sugar synthesis, that is glucose synthesis, occurs by only one pathway, that is C3 pathway. So C3 pathway, C3 cycle is the common biosynthetic pathway through which the synthesis of hexose, that is glucose, occurs. So it is present, C3 pathway is present in all types of plants, that is C3 plants, C4 plants and even in CAM plants, through which synthesis of hexose occurs. Okay, here what is happening? Carboxylation, that is carbon dioxide fixation, occurs two times, where once in bundle sheet cell, that is in first step, and once in mesophyll cell, and next second carboxylation is taking place in the bundle sheet cell. Now let's study about these carboxylations, first carboxylation and second carboxylation. So first carboxylation and second carboxylation. So where this first carboxylation is taking place? First carboxylation is taking place in mesophyll cell. First carboxylation is taking place in mesophyll cell, whereas second carboxylation is taking place in bundle sheet cell, that is in C3 pathway. Bundle sheet cell. Right. Okay. So this first carbon dioxide is fixed into what? It is fixed into PEP. So carbon dioxide fixation occurs into PEP, that is phosphoenol pyruvic acid, which is 3-carbon compound by the activity of what? Peptase enzyme. Peptase resulting in the formation of what? Oxaloacetic acid, which is C4 acid. C4 acid. So this is about first carboxylation which occurs inside the mesophyll cell. Next, coming to second carboxylation. So second carboxylation which occurs inside the bundle sheet cell is, so here carbon dioxide is fixed into RUBP. RUBP, that is ribulose 1,5-bisphosphate, which is 5-carbon compound, and this step is catalyzed by, second carboxylation is catalyzed by the enzyme Rubisco, that is RUBP, carboxylase, oxygenase, resulting in the formation of a product called 3-PGA, that is 3-phosphoglyceric acid, which is C3 acid. And hence what is this? This is C3 pathway which is taking place inside the bundle sheet cell. So this is about first carboxylation and second carboxylation. Okay, now let's come back to this pathway. So after formation of this C3 acid, the C3 acid is transported back into mesophyll cell from bundle sheet cell through plasmodesmatal connections, and inside the mesophyll cell, this pyruvic acid undergoes regeneration into phosphoenol pyruvic acid. So the step of regeneration is catalyzed by an enzyme phosphopyruvic dikinase. This phosphopyruvic dikinase enzyme is very special enzyme because it is sensitive to low temperatures. It is sensitive to low temperatures, and hence these C4 plants are adapted to tropical and subtropical regions. As this phosphopyruvic dikinase enzyme is sensitive to low temperature, these C4 plants, the plants in which C4 pathway occurs, are adapted to tropical and subtropical regions. Sensitive to low temperatures. Right. Okay, so during the regeneration of pyruvic acid, regeneration of phosphoenol pyruvic acid from pyruvic acid, 1 ATP. For the regeneration of one pyruvic acid, one phosphoenol pyruvic acid, 1 ATP undergoes splitting into, energy is consumed in the form of ATP, which undergoes splitting into AMP, that is adenosine monophosphate. So here AMP is released is nothing but here 2 ATPs are consumed. Why? Because if this AMP has to be converted back into ATP, the energy which is utilized by this AMP will be equal to the 2 molecules of ATP, and hence we can say that for the regeneration of 1 molecule of PEP, 2 ATPs are consumed. 2 ATPs are consumed. So totally for the regeneration of 6 molecules of PEP from 6 molecules of pyruvic acid, how many ATPs will be consumed? 12 ATPs will be consumed. So now let's study the energetics of this pathway. That means how many ATPs and how many NADPH are consumed for the fixation of one carbon dioxide and then 6 carbon dioxides or one glucose molecules, or for the synthesis of one glucose molecule through this pathway. Okay. So for the fixation, let's study ATP, number of ATP and number of NADPH consumed during this pathway. So during C4 cycle, for the fixation of one molecule of carbon dioxide, how many ATPs are consumed? 5 ATPs are consumed. 5 ATP. Why 5 ATP? During C3 pathway, 3 ATP. That we already studied in the previous video about this C3 pathway in detail. You just go through that video. Right. So during C3 pathway, 3 ATPs. Next, during the regeneration of PEP, 2 ATPs. So totally 3 plus 2, 5 ATPs for the fixation of one molecule of carbon dioxide. So for the fixation of 6 molecules of carbon dioxide or for the formation of one molecule of glucose, how many ATPs are consumed? 30 ATPs. 5 into 6. So 30 ATPs are consumed in case of C4 plants, that is in C4 pathway. Coming to NADPH. How many NADPH are consumed for the fixation of one molecule of carbon dioxide? 2 NADPH, where that is in C3 pathway, which is taking place inside the bundle sheet cell. So 2 NADPH. And for 6 molecules of carbon dioxide, how many? 2 6, that is 12 NADPH. Okay. So this is about C4 plants. Let's compare this energetics with C3 plants. So in case of C3 plants, for the fixation of 6 molecules of carbon dioxide or for the synthesis of one molecule of glucose, how many ATPs? 18 ATPs are consumed and 12 NADPH are consumed. So how many ATPs are consumed more in C4 plants, C4 pathway than in C3 pathway means 12 ATPs. Here 18 and here 30. And hence we can say that in C4 pathway, 12 more ATP molecules are consumed for the synthesis of one molecule of glucose. So now what is the ratio between the number of ATP molecules consumed during C4 pathway and C3 pathway means C4 30, C3 is 18. So 5 is to 3. Right? So this is about the energetics of this C4 pathway. And the C4 pathway is more evolved than C3 pathway. That means C4 plants are more evolved than C3 plants. Now let's study how they are more evolved than C3 plants in the next video. That is significance of C4 plants or C4 pathway. Hope you understood this concept well. Thank you, students.